So in the previous two videos, we described the structure of monosaccharides. In particular, we talked about the predominance of the cyclic structure in pentose and in hexose. In this next video, you will discover how monosaccharides assemble to form disaccharide and complex carbohydrate. Remember how a cyclic monosaccharide is made. An aldehyde function reacts with an alcohol group to form an hemiacetal, or a ketone group reacts with an alcohol to form an hemiketal. In the hemiacetal that is formed, you have now an hydroxyl group. This hydroxyl group can react with another alcohol to form an acetal. Or the hemiketal that contain a newly formed hydroxyl group can react with an alcohol to form a ketal. Now imagine that we start with a molecule of alpha-D glucose. It is an hemiacetal. An hydroxyl group has been formed, carried by carbon 1. If this hydroxyl group reacts with an alcohol, will form an acetal. If this alcohol is another monosaccharide, will then form a disaccharide. So let's take an example. Alpha-D glucose can react with beta-D glucose. So the carbon 1 carried by alpha-D glucose can react with the carbon 4 of beta-D glucose to form what is called a glycosidic linkage, or more exactly a 1,4 glycosidic linkage. We have now a disaccharide, which is called maltose. If we look in detail at the structure of this disaccharide, we can see that we still have a reducing group that is carried by the carbon 1 of the beta-D glucose unit. So this disaccharide will have a reducing end, and on the opposite side will have a non-reducing end. And for that reason, maltose is called a reducing disaccharide. So now we can form a large number of disaccharide by having a reaction between the anomeric carbon of one sugar and any carbon carried by another sugar molecule, as long as this carbon carries an hydroxyl group. So for example, we can have a disaccharide that is formed by the reaction of two identical monosaccharides. The carbon 1 of two alpha-D glucose can react with each other to form a 1,1 glycosidic linkage. And the disaccharide that results from this linkage is called trihalose. In this case, because the two reducing carbon of the glucose unit are engaged into this glycosidic linkage, the disaccharide is non-reducing. We can also have a reaction between two different aldose. For example, beta-D galactose can react with beta-D glucose. In this case, the carbon 1 of beta-D galactose will form a glycosidic linkage with carbon 4 of beta-D glucose. The disaccharide that is produced is called lactose, and it is a reducing disaccharide because it contains a reducing end corresponding to the carbon 1 in the beta-D glucose unit. Finally, we can also have a reaction between an aldose and a ketose. In our example, alpha-D glucose react with beta-D fructose to form a 1,2 glycosidic linkage. So the anomeric carbon of alpha-D glucose react with the carbon 2, which is also the anomeric carbon, of beta-D fructose. The product, the disaccharide, is called sucrose, and because the two reducing group of the sugar units are engaged in the glycosidic linkage, we'll have a non-reducing sugar. Now, the type of glycosidic linkage that we have described to form disaccharide can also form more complex polysaccharide, can form complex carbohydrates. And in the rest of this video, I will focus on one of these complex carbohydrates called glycogen. Glycogen is a polymer of alpha-D glucose and has a very complex branching structure. It is so complex and so large that we can visualize site of storage of glycogen when we observe section of skeletal muscle by electron microscopy. While most of the cells synthesize glycogen, glycogen is stored in two major types of cells, liver cells and muscle cells. Glycogen is in fact an intermediate short-term energy reservoir.
Does glucose in the muscle have the same purpose as glucose in the liver?
No. In fact, the glucose that is produced by the hydrolysis of glycogen in the liver will be exported out of the liver and distributed to other tissues that need a fuel molecule. While in the case of the muscle, the glucose that is released by hydrolysis of glycogen will be used in the muscle itself for its own metabolic needs. It will not be distributed to other tissues. Now let's look at the structure of glycogen. I said that glycogen is a polymer of alpha-D glucose. We find in glycogen long chains where the glucose units are linked with each other by an 1,4 glycosidic linkage. Then the complexity of the structure is increased by the addition of branches that are linked by a 1,6 glycosidic linkage. The consequence of this extensive branching is that for one molecule of glycogen, you have one reducing end, but numerous non-reducing ends, because each branch ends by a non-reducing end. When energy is needed, the breakdown of glycogen starts by the hydrolysis of non-reducing ends. This process, as well as its regulation, will be discussed in a later video. Let's focus now on the synthesis of glycogen. So when there is a high concentration of glucose in the bloodstream, tissues like liver and muscle will transport glucose inside their cells. And glucose will be converted into glucose 6-phosphate. It will be phosphorylated. In liver, the enzyme that catalyzes this reaction is called glucokinase, and in muscle, it's called hexokinase. These two enzymes catalyze the same reaction, but have very different kinetics, as we'll see later in this course. Once the glucose 6-phosphate is formed, it will be quickly converted into glucose 1-phosphate. And the next step will be a condensation reaction between glucose 1-phosphate and a nucleotide called UTP, or uridine triphosphate, to form a sugar nucleotide, UDP glucose, and pyrophosphate. This reaction has a high energy cost, but the extremely rapid degradation of pyrophosphate into inorganic phosphate drives the reaction toward the formation of UDP glucose. In UDP glucose, the molecule of glucose is activated, and UDP glucose will act as a donor of glucose unit. So an enzyme called glycogen synthetase will catalyze the transfer of a glucose unit from UDP glucose to the non-reducing end of a glycogen chain. The consequence will be the increase of the length of this chain by 1 unit and the formation of a new non-reducing end that can be used by the glycogen synthase to continue the elongation of this chain. Now, how are the branches formed? Glycogen synthetase do not catalyze branch formation. This reaction is catalyzed by another enzyme called the branching enzyme. This reaction starts by the cleavage of the end of a chain to form a fragment with 6 or 7 glucose units. The newly formed reducing end from this fragment can then form a glycosidic linkage, a 1,6 glycosidic linkage, with a residue that is contained into an acceptor chain. Then the glycogen synthase can catalyze the elongation of the newly formed branch, as well as elongate the branch that was cleaved off during the first step of the reaction. The continuous pruning of chains to create new branches and the elongation of these new branches create a very complex structure. On average, branches occur every 10 sugar units. At its maximum size, a molecule of glycogen contains about 55,000 glucose units and about 2,100 non-reducing ends. Now, I said that glycogen synthase can catalyze the transfer of a glucose unit to an existing chain. And that is because the glycogen synthase can only catalyze the elongation of a primer that is at least 4 glucose units long. Another protein called glycogenin is responsible for the synthesis of this primer. Glycogenin contains a tyrosine residue, and a glucose unit can be transferred from UDP glucose to this tyrosine. So the carbon 1 carried by the glucose unit can react with the hydroxyl group from the tyrosine residue to form an acetal. Once the glucose unit is covalently attached to glycogenin, glycogen synthase will form a tight complex with glycogenin. However, the synthesis or the addition of the next 3 units will be catalyzed by glycogenin and not by the glycogen synthase. Once the primer is assembled, then glycogen synthetase will take over, will elongate the primer, move away from glycogenin, and then will start to elongate the chain. The branching enzyme will come into play, and you will have the final organization of a glycogen molecule. So monosaccharides assemble to form disaccharide, to form complex carbohydrates such as glycogen. Glycogen molecule with their numerous non-reducing ends that are facing outward are very well suited for an easy mobilization of glucose unit when a cell needs energy very quickly. In the next series of video, we'll learn how glucose can be oxidized to produce ATP, the source of chemical energy that is used by cells to do work.